Students, today we are going to start very new, interesting, but recent branch of biology. We have to say that, or sciences, which is useful to you to understand the specific, the world that is genetic world of a plant. Here, genomics, the terminology first of all, is introduced in 1986 by the Tom Rodricks. Tom Rodricks is the name of scientist who has introduced the terminology genomics in 1986. My friends, I always used to say that the English peoples ruled to the world most of the country, including our country, isn't it? So we know that the English peoples have ruled to our country for 150 years. 150 साल. Only the principle they have followed, that is divide and rule. Similar way, I used to say that in case of science, if you divide this word genomics, isn't it? So genomics, that is genome is there. Then what is genome? Isn't it? Genome is nothing but the total DNA that present within the one cell of an organism. What is genome? Genome is nothing but the total genetic material present in the DNA or genetic material present within a one cell of an organism. That is known as a genome of that organism. Isn't it? Now, then that is about genome. So word genome. Genomics. What is that mix? Genomics is the study of structure and function of an genome of an organism. No doubt, genomics definition is very much clear to you, my friends. Genomics is the study of a structure and function of an genome of organism. And genome is nothing but the total DNA that present within the one cell of the organism. Genomics terminology introduced into the scientific world in 1986 by the scientist Tom Rodricks. Now, what are the methods of genomics? We have studied in the genomics, actually there is a study of a structure and functions of genome, that is known as genomics. In case of this method of genomics, my friends, you have to keep in mind there are several methods for sequencing of a genome. Several methods are there. Sequencing involves the determination of an exact order of the four bases for nucleotides on the DNA strand. So here is a sequencing of genome is nothing but we have to find out, determine the exact order, position, location of the four bases for the nucleotides on the DNA strand. DNA strand वरती असलेले न्यूक्लिओटाइड्स त्याची एक्झॅक्ट पोझिशन, लोकेशन या सिक्वेन्सिंग ऑफ जिनोम मध्ये आपल्याला अभ्यासता येते. Sequencing of genome chiefly comprises three steps. Sequencing of genome comprises three steps. The cloning of the DNA to be sequenced. ज्या डीएनएचा आपल्याला सिक्वेन्सिंग करायचं आहे त्याच्या अनेक कॉपीज तयार करायच्या, क्लोन्स तयार करायच्या, मेनी Xerox copy of that one. Clone is nothing but the Xerox copy of that one, isn't it? That we have to first of all produce in a first step. The sequencing reaction and electrophoretic separation. Sequencing reactions and electrophoretic separation with the help of electrophoretic technique that are to be separated, and analysis of a ensuing data. These are the three steps of sequencing of a genome is there. Now, are there chemical methods? Definitely. that one of the chemical may develop a maximum Gilbert in case of a restriction fragments of a labeled with 32 phosphorus at either ends of a 5 prime to 3 prime using either of the enzymes polynucleotide kinase or terminal transferase. From the reaction map, an enzyme is selected to remove of all the pieces from one end of the molecule, leaving just one, and label. The DNA is then chemically cleared at a specific residues in a 5 different reactions. These reactions are partially completed and partially digestion products are separated on a polyacrylamide gel and auto-radiographed that things. The fragments having the label of terminus are same. So chemical method is developed by the scientist, that is a maximum Gilbert, and maximum Gilbert have used a restriction enzymes, isn't it? And each and every after treating the DNA material genome to the restriction enzymes, which are fragments, that fragments are labeled either to its 5 prime end or 3 prime end. Now, the whole genome shotgun sequencing, another method of study of a genome, that is a sequencing of a genome, is a whole genome shotgun sequencing. J. Craig Venter and H. Smith, J. Craig Venter and H. Smith developed the whole genome shotgun sequencing and sequenced the two genome of a bacteria, Haemophilus influenza and Mycoplasma genitalium, isn't it? Now, here I will give you idea with the help of this diagrammatic representation what the things are going to happen. Here we will be able to see that genome is isolated, that is treated with the restriction enzymes, and we have found DNA fragments. That DNA fragments are spotted on a electro gel, isn't it? And when we will allow to pass through all these things, so what happens actually, the fragments of purification from the electrophoretic method, it is possible. So agar gel, the sheet is there. On this agar gel, electrophoresis of a fragments and DNA size markers are there. So separation of these DNA fragments which we have got by the treating or sonication. Sonication method is used here just to make pieces of a DNA fragment which is we have isolated from H. influenza, isn't it? Now these fragments again we have loaded on a agar gel, and then this agar gel sheet is electrophorized, and because of that one we have got the separation of a DNA fragment, then fragment purification from the gel. These are the purified structures are there. Clonal library preparation. Now each and every DNA fragment is cloned, xeroxed with the help of the further techniques and sequence the clonal inserts. Specifically, the end sequencer are there. End sequences are there, isn't it? Specifically the end sequences. Now end sequences we have got. Now construct the sequences, contigs and align using overlap, fill in the gaps is there. So this is a whole genome shotgun sequencing method. Steps I have explained to you very well. Try to understand all these things and how the DNA sequencing can be done here, that is to be understood by you with the help of this diagram. Sketch the diagram more and more time so that you will understand the exact methodology, that is a shotgun whole genome shotgun sequencing methodology. Now here the whole genome shotgun sequencing method, again diagrammatically we have seen here. It consists of four steps: library construction, random sequencing, fragment alignment, and gap closure and proofreading. These are the four steps of the whole genome. Shotgun sequencing methodology are there. Once again I will tell you: library construction, random sequencing, fragment alignment and gap closure, and proofreading. Now library construction. In case of library construction, chromosome is isolated from the desired cells. Chromosome, first of all, we have to isolate the desired cells. Following the methods of molecular biology, molecular biological methods are there. With the help of this methodology, we have to isolate the chromosome from the desired plant cells. The isolated DNA is randomly fragmented into small pieces. Randomly we have to make a fragments of that one, that could be into a small pieces using ultrasonic waves. With the help of ultrasonic waves, the isolated chromosomes from the desired cells are to be fragmented into a small pieces. Then the fragments are purified and attached to the plasmid vector. Then most method what you have to do, that is nothing but just to purify that fragments, then attach that to the plasmid vectors, and plasmids with single insert are isolated. A library of the plasmid clones are separated by transforming E. coli strains with plasmid that lacked the restriction enzymes. One thing you have to keep in mind, इथं आपल्याला काय करायचे आहे, वी हॅव टू कन्स्ट्रक्ट द लायब्ररी. कशाची लायब्ररी तयार करायची आहे, तर डिझायर्ड कॅरेक्टरिस्टिक असलेल्या डी एन ए फ्रॅगमेंटची लायब्ररी तयार करायची आहे. मग ही लायब्ररी तयार करत असताना काय करायचं आहे, डिझायर्ड सेलमधला जिनोम काढायचा, दॅट इज टू बी सब्जेक्टेड टू द अल्ट्रासोनिक वेव्ज, अल्ट्रासोनिक type of, yes, ultrasonic waves, make the fragments of that one, then purify that fragments which shows presence of a desired characteristics, and that are to be, we have to we have to integrate with the plasmid vector. After integration with the plasmid vectors, that plasmid vectors are to be inserted into a Escherichia coli, and then that Escherichia coli containing plasmid vector with desired gene having DNA fragment from the desired cell is to be introduced into E. coli, and that E. coli then we have to multiply in the in vitro conditions. But we have to select such a type of E. coli which could not be able to damage or cut or destroy the desired gene containing DNA fragment and plasmid of the vector. Random sequencing is there. The DNA is purified from the plasmid. The thousands of DNA fragments are sequenced by using automated sequencer, by using of a primer labeled with the special dyes. Normally with the universal primer, thousands of templates were used. These recognized the plasmid DNA sequence, next bacterial DNA insert. The whole genome is sequenced several times. Sequencing is not possible with the help of a single attempt. Many attempts we have to make, many times we have to multiply, many times we have to automate that one, automated sequencer we have to take help. Then only it is possible the sequencing of genome, sequencing of DNA material. And this method, that is a random one. Now fragment alignment and gap closure is the another step. Fragment alignment and gap closure. By using a special computer program, the sequenced DNA, by using a special computer program, the sequenced DNA fragments are clustered and assembled into a longer stretches of a sequence by comparing the nucleotide sequences overlaps between the fragments. The two fragments are joined to form a large structure of DNA if the sequence at their ends overlapped and matched. This overlap comparison method resulted in the set of a large nucleotide sequence called as a contigs. Contiguous nucleotide, that is known as a contigs. The contigs are aligned in a proper order to form a completed genome sequence. To form a completed genome sequence. Now proofreading. We know that after doing all these things, we have to finalize the things. Before finalizing, we have to read the proof of that one. Then the proofreading of sequences is done carefully so that any ambiguity in the sequence could not be resolved. The sequences are also checked for the presence of any frame shift mutation. If so, the mutation is corrected. If you found any difficulty, any ambiguity in the sequences, then what we have to do? So we have to actually correct that ambiguity at this. That is nothing but the proof reading is there. Now, what are the types of genomes? We have studied the genome. Genomics is the one of the terminology that is nothing but the study of a structure and functions of the genome of the cell. That is nothing but the genomics. There are three types of the genomics. Functional genomics. Here, in case of functional genomics, what happens actually? Identification of the genes and their respective function is that. This is one of the type. Genomics is most important type of a functional genomics is most important type of a genomics, and this type actually includes the identification of genes and their respective functions. Another type, that is a structural genomics. Here the prediction related to the functions of the protein. Prediction in structural genomics, we have to decide one of the structure that will help us to predict the exact function of the proteins which is not known to prediction methodology. So prediction is there, that is a structural genomics. Comparative genomics, for understanding the genomes of a different species of organisms, so we can compare the functions, characteristics are followed by A species and compare that with B species characteristics followed. So understanding the genomes of a different species of organic possible with the help of third type of a genomics, that is a comparative. So there are the three types of the genomics: functional genomics, structural genomics, and comparative genomics. Functional genomics is nothing but the identification of genes and their respective functions. Structural genomics useful to predict the related function of the protein. Comparative genomics, that is for the understanding the genomes of a different species of organisms. Now the applications of the genomics, very important thing is that of the genomics can be used in industry to identify the novel enzymes. Very useful genomics uh, is there. With the help of the genomics, we will be able to identify the novel enzymes which are useful to the industry. This has a potential to enhance the bioremediation. Keep in mind, bioremediation in the sense we know that many more organisms are there. They are having a capacity just to remove the hazardous waste and improve the techniques for the microbial production of a methane and other fuels. So my friends, we know that most of the things are there. Pure things are present there, but that pure things cannot be extracted. And in such a cases, what we have to do? We have to use the bioremediator. So bioremediation of a hazardous waste is possible by getting idea by studying the genomics very well. Genomics has application in production of DNA chips. When we are interested to produce the DNA chips, it is possible. These DNA chips are used to diagnostic and genetic mapping. My friends, most of times we cannot find out where is the exact problem. So uprooting of the problem, that is the exact and right correct 100% solution of the each and every problem. Uprooting, मुळापासून उपटून काढायचं. Suppose we are having a problem in our hand, but we cannot find out the exact where is the problem. So up to that point, that root is possible to reach with the help of these DNA chips. So DNA chips have been prepared to identify the mutation, multiple mutation, genotyping of hepatitis C virus in the blood samples, actually where that virus is creating problem in hepatitis C. So that Once we come to know that, it would be a very easy. Diagnosis is most important. Without diagnosis, the patient cannot be treated, isn't it? Not only by external seeing, it is very difficult. So once we diagnose the disease, then it is possible to develop the again medicine on that one, although that disease is a new one that we can see, we can observe, we can experiencing in case of this pandemic COVID-19. Okay, so that is possible with the help of a genomics. Another thing is that genomics has application in agriculture biotechnology. Expression patterns of agriculture crops or transgenic plants under the different environmental conditions can be analyzed by using DNA chips. Suppose we have developed one of the transgenic plant, the plant containing foreign gene having a desired characteristics, but expression of that character, expression of that gene is possible to understand under different environmental conditions by using DNA chips technology. It is also used to study the DNA polymorphism when the molecular markers are associated with the selection is to be studied. Genomics can provide insight into a pathogenicity and suggest the treatment for the infectious diseases. Very dangerous, hazardous diseases are there which are creating problem to the life of the plant, and definitely that leads to the decrease in the yield. So actually the genomics technology will provide us a very interesting point is that we will be able to find out the insights into biopathogenicity, and it will again help to suggest the treatment of infection, treatments for the infectious diseases. Now, virulence genes can be identified and its expression during the infection along with the host response can be studied. More sensitive diagnostic tests, new vaccines and antibiotics may come from the genomic study of the pathogen. My friends, you will have to keep in mind these two words, these lines having a lot of importance and use of that genomics branch to us. If we study the genomes, then it is very easy to find out the exact diagnosis test, find out the exact diagnosis test. New vaccines can be developed, new antibiotics can be developed very easily and very properly and in a short span of time. So that is possible only with the help of a genomic study. Animals and plant genomics or genetics play significant role in vaccine and therapeutics development, breeding and selection of a meat quality, milk production, selection of a meat quality and milk production, and pest resistance capacity. So you'll have to keep in mind all these things are possible with the help of just to get idea. Now here, genomics will positively affect the agriculture. It may be used to find out the new biopesticides and improve the sustainable agriculture practices through the enhancement in the process such as a nitrogen fixation, phosphate solubilization, and production of a indole acetic acid and HGN, etc. My friends, we know that most of the plants are unable to synthesize their own nitrogen requirement or complete their own nitrogen requirement, although they have been swimming in a sea of a nitrogen, 78% nitrogen about that, but they are not unable to get that one. That is not available to for their growth. In such a conditions, we can make plant to fix atmospheric nitrogen. We can make a plant to themselves absorb the phosphate or solubilize the phosphate which is available around the root zone of their plant. So that is possible with this identifying the genes and gene products in the organ that lead to the diseases in the human will lead to the development of a treatment for these diseases. In the sense, suppose one of the organism infect get in infect we have to say that infect to the human being, but which gene of that organism is a secreting toxic substance or a such a substance which causes disease to the human being, that we can identify with the help of genomics. And once we could be able to identify that gene, we can find out antitoxins, we can find out antibiotics, we can find out the vaccine just to nullify the effect of that specific secreted structure, isn't it? So here very interesting thing is that this genomics is going to help to make the human or protect the human from many more diseases. Students, today we have got idea about in this genomics topic, that is the what is genomics, what is genome, what are the types of genomics, isn't it? What are the applications of genomics, isn't it? Also, we have learned the DNA sequencing methodologies, isn't it? So that you have to understand very well, keep in mind. And if you have any difficulty, just to do, bring to my notice. With this, thank you very much.